Okay, good evening once more. So welcome to Metabolic Chemistry, and this is going to be an interesting journey for all of us, and we're going to enjoy because we there will be need for us to read and read and read, memorize pathways. Okay, we'll be sleeping on pathways now. Okay, so I welcome you to Metabolic Chemistry, and this is medicine. Okay, this is medicine, and this is an overview. So we want to talk about everything in here. We'll talk about each and every pathway. We'll give an example of each and every pathway that happens in the human body. Okay. So this part of the course is very important because it's the one that is going to tell you what kind of metabolic condition a person is suffering from. Okay. So most of the endocrine patients, they have got metabolic inborn error of metabolism, or they can acquire the metabolic errors. Okay, so what is metabolism? So in this class, by the end of this class, students should be able to understand what metabolism is and its connection to medicine. Okay, so remember that we are dealing with enzymes this time around. Okay, so it's either a pathway is biosynthetic or it's catabolic. Okay, so a pathway can either be biosynthetic or catabolic, meaning you're making something from the building blocks, or you're breaking down a complex compound um, into its monomers. Okay, so for you to understand diseases like albinism, you need to be able to understand the biosynthesis of melanin. Now, the question is, we have two things in metabolism, catabolism and anabolism. So it's either we are making or we are breaking. Okay? In this case, we are making. And we are using enzymes. So these pathways, they use enzymes. Okay? Don't forget your structures, and don't forget your enzyme classification and catalysis, please, because you need the integration of that knowledge to be able to memorize these pathways. Because when you're memorizing a pathway, it means that you're memorizing one, you are memorizing the intermediates, two, you are memorizing the enzymes, three, you are memorizing the products. Okay, so each pathway can either be an anabolic pathway or a biosynthetic pathway, which is, sorry, anabolic is biosynthetic, or it can be catabolic pathway. Okay, so for example, if you want to understand albinism, you need to understand the biosynthesis of melanin. So melanin is a skin pigment. Okay, even in the eyes, melanin is also needed. That's why these people who are suffering from albinism, they can get blind because they are photosensitive. But what's the problem with the albinos? Eliza, would you mind telling us what you think is the problem with albinos? What's really the problem? Hello?
I think they have a problem with the production of their melanin.
Yes, they have a problem with the production of melanin. And melanin, the precursor, if I say precursor, I mean the compound which you need, the first compound needed to make that particular um, organic compound that you are looking for. So if you want to make melanin, you need an amino acid. And this amino acid is called tyrosine. Okay? So tyrosine is an amino acid required for the biosynthesis of melanin. Now, the first two steps are critical to the synthesis of melanin. And these first two steps, they are they are characterized by an enzyme called tyrosinase. Okay, now these people who are suffering from albinism, they are deficient in the enzyme tyrosinase because of defective genes coding for this enzyme. Okay, lacking this enzyme, it means that you are not going to produce melanin. Okay, and your skin will appear white. Okay, so those people we know whites from England, those are not albinos. Okay, those ones are just normal people. They have melanin, but it's not a lot. But albinos, they are completely don't have melanin. Okay, and melanin is very important on the skin and also in the eyes. Okay, other membranes. Okay, like in the eyes. So now, this enzyme tyrosinase is actually a molecular target for a group of drugs that we call hydroquinones. Now, what are hydroquinones? Um, Nivea, do you know Carolite? Do you know
No,
Carolite?
sir.
Have you ever used Carolite before? Have you ever
No,
used
but
Carolite before?
no, I just know that it's a lighting cream.
Okay, now how do you think it works? What's in there? What drug is there? So we have hydroquinones in there. So there's a drug called hydroquinone. Okay, these ones are tyrosine inhibitors. Okay, and these are skin lightening agents. So without understanding biochemistry, you will never understand anything to do with metabolism in the human body. And we can't have such a doctor who doesn't understand metabolism. Okay, because it has got clinical implications. Almost all endocrine patients, you need to understand um, metabolism. Okay, otherwise you are never going to treat any endocrine patient. So hydroquinone is a drug. It's a group. It's actually a group of drugs that blocks the enzyme tyrosine uh, kinase. It's actually tyrosinase or tyrosine kinase. Okay, so tyrosinase is the enzyme required for the synthesis of melanin, meaning this person is not going to be producing melanin. It's blocked. Okay, the pathway for producing melanin is blocked. If they are producing, it will be minimal. Okay, because the enzyme is blocked. And you guys, you are experts. You understand those things of reversible and irreversible inhibition, and also the whether you're binding on the active site or you're binding on the allosteric site. So you understand that this enzyme is actually an allosteric enzyme, meaning it is the one that determines the rate at which melanin is produced. Okay, so hydroquinone is a drug that is found in those lightening creams. Please take note of this. Okay, so tyrosinase inhibitors are drugs that are used in making the skin to become light. Okay, so this is what happens in albinism. The person doesn't have tyrosinase, it's just deficient. Okay,
Sir, I have a question.
yes, please.
What about these people? Let's say they were black, well, not like they were producing melanin. Then all of a sudden they stop producing melanin. Like these people who we see they are like they are developing the skin of an albino. What happens to those people?
I've never seen such a person before. Where have you seen that person?
Around, like maybe they are having marks like bamba, like not like we Vales Bamafta. Like someone has just started having that skin of an albino, maybe in patches.
You mean the skin is starting to become hypopigmented?
Yes. What happens to them?
Maybe she's
Oh,
talking about
oh,
vertigo.
oh, she's talking about vitiligo. That's an autoimmune condition. It's autoimmune. So, you know what autoimmune conditions are, where the immune system attacks the what? it attacks the something. So in this case, attacking the receptors where um, melanin is supposed to bind, because melanin is a hormone. Remember, okay, melanin is a hormone, and it has to bind somewhere, okay, for it to produce its effects. So autoimmune conditions are going to attack those receptors where melanin is supposed to bind, meaning you have the melanin, but it's not doing anything. Okay, you get it?
Yes, sir, I get.
Okay, that's good. So, guys, you need to be happy. Metabolic chemistry is the art of medicine because you need to understand the pathways properly. So, whatever we have done, carbohydrates, lipids, nucleic acids, okay, all those things, him, okay, all those things, they have got biosynthetic pathways. They also have catabolic pathways. Okay, those are the things that are in the syllabus. That's the syllabus. Okay, so the food that we know is actually the syllabus. Carbohydrates, okay, lipids, nucleic acids, proteins. That's the syllabus. Okay, so we are going to start with we are going to start with metabolism of carbohydrates. That would be like it's a component. It's not a topic. It's a component, and inside it we have a lot of topics. Topics like the glycolytic pathway. So next week we'll start the glycolytic pathway. Okay, we'll start the glycolytic pathway. So the glycolytic pathway we have gluconeogenesis, we have glycogenesis, we have glycogenolysis. Okay, this is under carbohydrates. Okay, we have the citric acid cycle, we have the electron transport chain. Okay, and these are very high yield topics in the exam, so you can't afford not to understand what's going on here. We need people when they're dreaming about pathways. Okay, then from there we'll go to the lipids. We have lipid biosynthesis, we have cholesterol biosynthesis. You see how the human body produces testosterone, for example. Okay, how it produces the female sex hormones, the estrogen and the progesterone. You see how the human body can synthesize the heme. Okay, you see how the human body can synthesize those nitrogenous bases that you learned a few weeks ago, even in first year you learned in Bio 1400. You see how the human body can make those rings. Okay, so the major site of biosynthesis or metabolism is the liver. The liver is the major site. That's the reason why most of the things that happens they have to first happen in the liver, and most of the drugs they are targeting the liver, so they have to first undergo through they have to go through the liver for them to be activated. And you will never understand pharmacology and therapeutics if you don't understand metabolic chemistry. You will never understand pharmacology and therapeutics, and you cannot be a medical doctor who doesn't know drugs. Okay, for you to understand how drugs works, most of the drugs that we have, their molecular targets are enzymes. Okay, their molecular targets are enzymes, and in this course we are going to teach you the enzymes you are supposed to enjoy. So welcome to metabolism. It is an interesting journey, and I hope students are going to enjoy it. Okay, so it actually has two tests because it's very long. So in this same metabolism, you have test one, you also have test two from the first part, then test three. In fact, you must have four tests. Okay, so guys, you must be free to ask because the students who are just from leaving, who are who are your immediate seniors, these ones they were complaining too much about the pathways. Okay, and you can see when you just get the results that Senate produced last academic year, you can see that PGY 2310 killed a lot of students. It's because of this same part, the metabolic part, because it requires you to memorize the pathways in your brain. Okay, God gave you a brain for a reason, so you're not going to complain. So why do I keep saying pathways, pathways? What's a pathway? A pathway is a route through which metabolism can take place. Okay, either it's synthesizing something or it's breaking down something. Okay, synthesizing things in the human body, it doesn't just happen like in one step. Even organic chemistry, you will see that you have got mechanisms. Step one, step two, step three, step four. This is also the case, but in organic chemistry, they don't talk about enzymes. Here will tell you enzymes. Step one is going to catalyze. For example, I'll give you an example of heme biosynthesis. Okay, I'll give you an example of heme biosynthesis. So, what's the clinical implication of heme knowing the heme biosynthetic pathway? Oh, I'm not moving the slides, I'm just explaining things. So, what's the implication of knowing the heme biosynthetic pathway? So, heme, you know, it is found in hemoglobin, and you know the function of hemoglobin. Nivia, what's the function of hemoglobin?
To transport carbon dioxide and oxygen in the blood.
Yes, so
And
it
then to
is.
carry.
So it is required for the for carrying oxygen and also carbon dioxide in blood. Okay, so very important to understand how heme is produced. Why? Because if a person has got anemia, for example, they are deficient in the um, eh, they are deficient in the hemoglobin. That is anemia, and they can die because anemia kills. Okay, anemia kills because it is going to affect the cardiovascular system. So now, listen. This is a pathway and it's a biosynthetic, meaning it's anabolism. Okay. So this pathway, it has got a precursor molecule. A precursor molecule is the first molecule that you see, first molecule that you see. I think I need to include this in the notes. So I'll stop presenting because I want to include this in the notes. I won't take long. So I'm not taking long. I just want to include this in the notes. It's a good example for students to learn on. Okay, let me just include this picture in the notes. You can be asking me questions as we are as I'm putting the slides. Eliza, you don't have questions for me?
Sir, may I have question?
Eliza,
Interesting.
yes, Mkata.
Oh. Yes. I have come across a words like biological reactions
Mm-hmm.
to,
Yes.
to differentiate it for me with the metabolic reactions. What's the difference between biological reactions and metabolic reactions?
Metabolic reactions, it's the same thing as biological reactions. There's no difference. There's no difference at all. I'm here to explain what metabolism is. So I haven't talked about anything to do with today's the slides that are there that people are supposed to be studying. Okay, so I'm putting the this part in your just needs to come second to this set. So first thing you need to see, and students should never forget this pathway because it's the first thing you have seen. in biochem. Okay. Oh,
What is that chemical that is found in chloride that you said? I forgot.
hydroquinone. Hydroquinone. It's a drug. Okay, it's a group of drugs. Okay, this is nice. So I'm just zooming in this so that people can see, so that we don't lose focus. Okay, I think we are good to go. You can go back presenting. So you need to enjoy this. You need to enjoy this. You must enjoy today's class. Okay. So, so what is metabolism? Because you need to understand what metabolism is. Okay, you already know what biochemistry is. Now, what is metabolism? So, metabolism is the sum total of anabolic and catabolic pathways in the human body, because we're dealing with the human body, not animals. Okay, so this is the sum total of anabolic and catabolic pathways. So, anabolic pathways, these are going to make something. They are also called biosynthetic. The catabolic pathways, these are breaking down molecules. Okay, for example, we can break down glycogen to produce glucose. Okay, we can break down glycogen to produce glucose, and we can make glycogen from glucose. So, making glycogen from glucose, is it anabolism or catabolism?
It's
Hello?
anabolism, sir.
It's anabolic.
Very good. So, guys, I need students to have at least you have a B plus in PGY 2320. Guys, this is nice, and you can take interest in this because we have so many diseases on the wards. Huh? I think one of these times I'll be taking students to the ward to go and see these metabolic conditions so that you can get to understand what we are talking about. Okay, are we together?
Yes, doc.
Okay, so you need to love this. This must be the love of your life, guys. Okay, not these human beings you put your trust in. This must be bread and butter for you. You need to enjoy this. Right. So when you're making, for example, you're making vitamin B3. So vitamin B3, who remembers the scientific name of vitamin B3? Eliza, do you know the scientific name of vitamin B3? Eliza, do you know the scientific name for vitamin B3?
No, sir, I've forgotten.
You used to know it. Who knows the scientific name for vitamin B3?
Just remember it starts with an
Livia, do you
N.
know it? It starts with what?
Letter N.
Yeah, it starts with an A. Nivea. Do you know the weight?
Is it nicotine? I'm not
No,
sure. I've forgotten.
no. Come again. Try again.
I
Emmanuel,
think
are
it's
you are you in
Alternate
niacin.
class?
spelling, sir.
Niacin.
Uh, oh, guys, come on, guys. Okay, so I'm going to do this. I'll write the weight. Okay, let's stop presenting because we want to see what will be written here. Stop presenting. Okay. Okay. So we have vitamin B3. Okay. Can people see this?
Yes, sir.
Okay. So vitamin B3 is called niacin. Okay. So niacin is very important in the normal skin architecture. Okay. Normal skin. You need niacin. If you are deficient in niacin, you will suffer a disease called pellagra. For a medical doctor to diagnose this disease, they need to understand the biosynthetic pathway of niacin. So niacin is vitamin B3, and the precursor for the biosynthesis of niacin is an amino acid called. Can someone guess? Who wants to guess? Which amino acid makes niacin or vitamin B3? From which amino acid does niacin come from? Toera, do you want to try? Or Gretson, do you want to try? Gretson, do you want to try?
No,
Hello?
sir. No, sir.
You don't want to try?
But I think it should be tryptophan, something like that.
Wow, this guy is studying. How did you figure it out? It's tryptophan. Hmm. This man is studying. Wow, I'm impressed. So, tryptophan is the amino acid required for the synthesis of niacin. There's a transamination reaction. Do you remember the transaminases? Those enzymes required for the transfer of certain groups other than hydrogen. Do you remember them from enzymes? Do you remember the transaminases or the transferases?
Yes.
Which group or which class of enzyme are they? Is it class 1 or class 2 or class 3 or class 4? It's class 2, okay? So the transaminases the transferases, they belong to class 2. Okay, right. So if you drink beer, we have someone who drinks beer here or abuses alcohol. Okay, so alcohol. What happens in pellagra? Alcohol. Alcohol is a culprit. Okay, so alcohol depletes. It depletes cofactors. Okay, things that are required for you to for the enzyme, the transferase enzymes to catalyze the formation of niacin from tryptophan. And the person who is deficient in niacin, it means they'll suffer from pellagra. So don't worry, there's a topic called vitamins. We're going to do all these things. So for here, just know that this is anabolism. Okay? So anabolism is where you're making something. In this case, we're making niacin. So if you're going to make glycogen, for example, glycogen should come from glucose, right? This is what you know. This is the normal thing that you know. But you can see glucose to glycogen, this is not one step. There are a lot of things going on here. There are a lot of enzymes going on here. And this pathway is called glycogenesis, because genesis means you make something from the scratch, just like God made the universe from the scratch. So glycogenesis is an anabolic pathway. So if you see a topic on the course outline written glycogenesis, that is an anabolic pathway. So you should know in your head that you were making glycogen. Okay, we also have another pathway called gluconeogenesis. Please, you must be able to practice how to pronounce these names, these words. We don't want to see students insulting. Okay, Toela, I don't want to see you insulting. So you must be practicing these words, how to say them. So gluconeogenesis, is it catabolic or anabolic? Anyone to try? Anyone to try? Is
Guess
gluconeogenesis anabolic or catabolic? Tracy, is gluconeogenesis catabolic or anabolic?
I think it's catabolic.
Okay, so Tracy thinks it's catabolic. Let's see the views of other people.
Anabolic.
Elisa says it's anabolic. Pauline, what's your say?
Anabolic.
Pauline says anabolic. Tony, what's your say? What do you think is gluconeogenesis? Is it catabolic or anabolic? Because a pathway should either be anabolic or catabolic.
I think it's catabolic.
Okay, Tony thinks it's catabolic. Fair enough.
I also think it is catabolic.
So you said glucogenesis or glycogenesis?
Gluconeogenesis. Here, it's here.
Gluconeogenesis. Oh,
It's
I
here.
think it's catabolic.
Okay, she's changed her mind. Who is for anabolic? Raise your hands. Those who are for anabolic, raise your hands. So Great Son is for anabolic. Who else is for anabolic? Who else is sticking to anabolic? Nivea as well. By the way, Pauline as well. By the way, those who have tried to watch the recordings where we are opening the camera, does it show? Are people able to see that the camera is opened? Anyone who has?
Yes.
Oh, okay, that's good then. If it's showing, I'm glad. Okay, so gluconeogenesis again. Genesis means you make something from the scratch. I gave an example of God making the universe. So gluconeogenesis is anabolic and not catabolic. Catabolic means you are breaking down something, you are not making something. Okay, so is glycogenolysis catabolic or anabolic? Glycogenolysis.
Catabolic.
Okay, Pauline says it's catabolic. Who else is saying it's catabolic? Tony, is is glycogenolysis catabolic or anabolic?
I think it's catabolic.
Okay, so Tony thinks it's catabolic, which is a good thing. Okay, those who are thinking it's anabolic, raise your hands. Those who are saying that um glycogenolysis is anabolic, raise your hands. Those who thinks that glycogenolysis is catabolic, raise your hands. So everyone says it's catabolic. Yeah, that's true. Lysis means breaking down something. Okay, so you're correct. We are on the correct route. So, as I said, we have anabolism plus catabolism. So, anabolism plus catabolism is equal to metabolism, which means metabolic chemistry will be comprised of both anabolic pathways and catabolic pathways. Okay? So I'm preparing your minds. Where you have come, you need to be memorizing enzymes. Okay? You need to be memorizing enzymes. Students who are new, please catch up because you need the structures. But let me be clear on this one. Metabolic chemistry, we don't care about the structures. Whether the student knows how to draw the structure or not, we don't care. And you should never draw a pathway with structures. Okay, we don't care about the structures in metabolism because our assumption is that the student already knows what is going on. You already know the structure of glucose, you already know the structure of fructose, so there's no need. When you're drawing a pathway, just know that you need to be mentioning, just writing words is enough, right? Glucose is converted to glucose 6-phosphate. Don't draw the structures. I hope this is clear because I had problems with biochemistry students last year. They kept asking me, are we supposed to know the structures on metabolism? No. No, 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 no. Is it clear? Wanda, is it clear?
Yes, it's clear, Doc.
Okay. So we need to enjoy this. We're here to enjoy. We're here to study. We need to enjoy this. Okay? And if you're not going to understand this course, it means that pharmacology next year will be bad for you. You'll be failing to understand how drugs works. Okay? And we have a lot of drugs, huh? We have a lot of drugs, and their molecular targets, most of them are enzymes. Okay, this is not magic, it is just biochemistry. Okay, now let's go back to the slides. Okay, so people now understand what catabolism is, they understand what anabolism is. Okay, so there's one pathway which I need you to know. I actually mentioned this when I was introducing students to the tuition center. So this is the pathway. People are able to see these words that are written here. Hello?
Yes, a bit, yes.
Okay, so you'll be able to see because I'll be explaining what these things are. So this one is a pathway, and it's an anabolic pathway. You see, it looks very complicated, but then it shouldn't be the case. Okay, so whatever you're seeing here are substrates, you are seeing intermediates. Okay, what is this? This is the mitochondria. So compartmentalization is very important in metabolism because pathways they are compat they are they do occur in compartments. Okay, so this is the mitochondria. So most of you know that in the mitochondria there is energy being generated, ATP. It's a powerhouse. That's what you know it of. But we have other things happening at the same time. Okay, and this pathway is called the heme biosynthetic pathway. It's an anabolic pathway. So don't worry about the details of this because we'll come back to this. This was just for you to understand that this is anabolism. Now, for you to make heme, that same heme which is found in hemoglobin, you need an amino acid called glycine. You also need another compound called succinyl coenzyme A. Okay, so succinyl coenzyme A is going to react with glycine in a reaction which is catalyzed by an enzyme called ALA synthase. Now, ALA synthase can be inhibited by the end product heme. So meaning you can control this enzyme. Once you produce your heme, it can regulate how this enzyme is working. Okay, you learn about enzyme structure and classification. You understand what's going on here. So this is an allosteric enzyme. Out of all these enzymes, this is a allosteric, meaning it regulates the rate at which the product is formed, the end product, not just a product, but end product. So this pathway is divided into two. We have got 4 reactions happening in the mitochondria, and we have got 4 other reactions happening outside the mitochondria, which is the cytosol. Okay, so for you to synthesize heme, you need glycine. Okay, glycine is a non-essential amino acid, meaning it is always there in the body. Okay, it is always there in the body because it needs you to make the heme. Okay, so glycine is going to be condensed with succinyl coenzyme A to produce ALA. So ALA is gamma amino levulinic acid in full. Okay, so now since the intermediate product for this reaction is ALA, look at the name of the enzyme, ALA synthase, which means this is good for the student. Okay, this is good for the student because you'll be able to remember that the compound which is formed is ALA, meaning the enzyme is ALA synthase. And you guys, you know what a synthase is. You know what a synthase is and the difference between a synthase and a synthetase. You must be able to tell me the difference between a synthase and a synthetase. So ALA synthase is an enzyme which catalyzes the condensation of glycine to succinyl coenzyme A to produce gamma amino levulinic acid, which is called ALA. So this enzyme needs PLP. Okay, what is PLP? PLP is pyridoxophosphate. Okay, pyridoxophosphate. Without pyridoxophosphate, this enzyme allows synthase cannot work. So meaning, what is pyridoxophosphate? Pyridoxophosphate is vitamin B6. A person who is lacking pyridoxophosphate, they will suffer from anemia. And if you don't understand how the anemia come about, it means you don't know this pathway because this pathway uses vitamin B6 as a cofactor for this major enzyme ALA synthase. So once you produce your ALA, this ALA it is produced in the mitochondria, so it has to diffuse through the membrane of the mitochondria via a shuttle. So a shuttle is just a molecule which is transporting this particular intermediate from one end of the membrane to the other. So a shuttle is going to transport ALA to the other side of the compartment, which is the cytosol in this case. So you have your ALA here, and then once the ALA is formed, it has to undergo dehydration via an enzyme called ALA dehydratase. So you can see the enzyme is telling you what is going on. So what is going on here is dehydration. Dehydration is the loss of water. So dehydration of ALA is going to produce porphobilinogen. Now, this porphobilinogen is actually a porphyrin compound. That's why when I tell you that the porphyrin rings they are found in heme, you shouldn't be shocked. You can see porphobilinogen. So porphobilinogen is actually a porphyrin molecule. Okay, and people, if you have seen people suffering from some skin conditions, we have a skin condition called porphyria, porphyria, porphyria cutanea tardae. So this disease is where a person is deficient in the enzyme called uroporphobilinogen synthase, meaning this guy is not able to push the porphobilinogen to become hydroxymethylbilane. So porphobilinogen has to become hydroxymethylbilane via the enzyme urod. Is it urod? Yes. So this is uroporphobilinogen 1 synthase. You need this enzyme to convert porphobilinogen to hydroxymethylbilane. And this one is a deamination reaction. Okay. So you follow the pathway, okay? So from ALA we form porphobilinogen, and you need an enzyme called ALA dehydratase. Then once you have your porphobilinogen, have I been kicked out of the meeting? No? Just okay. So oh, okay, so we have our hydroxymethylbilane, which has been formed from porphobilinogen. So now this hydroxymethylbilane is supposed to undergo further reactions. So from here it is going to become uroporphobilinogen. Okay, so uroporphobilinogen 3. Okay, via the enzyme uroporphobilinogen 3 uh, co-synthase, sorry. So once you have your uroporphobilinogen 3, it has to undergo decarboxylation, oxidative decarboxylation via the enzyme uroporphobilinogen decarboxylase. You can see the name of this molecule. You put decarboxylase, it makes the enzyme, and you're going to produce coproporphobilinogen 3. This molecule has to diffuse through the membrane of the mitochondria to go back, because the enzyme which is required for further reactions, it is found in the mitochondria, so it has to go back. Okay, and the reason why ALA is leaving the mitochondria, because these enzymes are they are not found in the mitochondria. So in this case, you can see we have um, coproporphobilinogen 3, which is going to be converted to protoporphobilinogen 3 via the enzyme coproporphobilinogen oxidase. Okay, so coproporphobilinogen oxidase is an enzyme that is going to convert coproporphobilinogen 3 to protoporphobilinogen 3. So from copro to proto, but the same molecule. Once you have your protoporphobilinogen 3, it will undergo another oxidative reaction where it will be converted to protoporphyrin 3. Okay, so it is no longer protoporphyrin 3. So it is now protoporphyrin 3 via the enzyme protoporphobilinogen oxidase. So you can see the name of this molecule, you put oxidase because you need to understand that this reaction is an oxidation reaction. from here we have oxidation, another oxidation. Okay, so once you reach protoporphyrin 3, it has to be converted. Now the final step is to convert protoporphyrin 3 to heme now, and the enzyme required for this is called phylokerotase, which is also called heme synthase. So heme synthase is an enzyme which is the final enzyme required for the production of heme from protoporphyrin 3. Now we have a group of diseases called porphyrias. You can see the word porphyrin here. So porphyrias, the result we have deficient in these enzymes. Okay? We have a condition called porphyria cutaneous tada. So this one is a deficiency in the enzyme uroporphyrin synthase. If you are lacking uroporphyrin synthase, no, uroporphyrin decarboxylase, you suffer from porphyria cutaneous tada, where you have increased uroporphyrin and also other porphyrins in blood, and this they get to be deposited on the skin, and then they will appear like reddish. You just check this disease out, you see. So the enzyme lacking here is uroporphyrin decarboxylase. Right. Now, if you have seen, if you remember what I wrote in the group in December last year, I was talking about lead poisoning. So lead poisoning affects the enzyme called ALA dehydratase. Okay, so ALA dehydratase is an enzyme required for the conversion of ALA to porphyrin. So for you to convert ALA to porphyrin, you need ALA dehydratase. Okay, so what happens in lead poisoning? So people are coming from Kawe. Most of them, they are very down. Why? Because this enzyme is blocked in lead poisoning. So ALA dehydratase is going to be blocked by the lead. Okay? So once this enzyme is blocked, it means you can't form heme. You can't form heme. If you can't form heme, you appear, you have anemia. Okay, so this will be, you be anemic. Okay, you'll be pale, and that's a disease. Okay, you need to be treated. So heme will be deficient as well in you, because you are not able to synthesize heme, because a large dehydratase is required and it is blocked. So these people, they have accumulated ALA in the blood, and this accumulated ALA in the blood is going to go to the brain, enters the brain. Once it is inside the neurons, it is going to attract water to it, and then the neuron is going to swell, and then they'll start dying one by one. That's the reason why you've got neurocognitive dysfunction in people who have got lead poisoning. Go and check up those kids who are in Kabwe. They're not doing well, okay, because of increased ALA in the blood, which goes to the brain and destroys everything. Okay, so this pathway is not meant for you to memorize at this point. What you have to know is that I've taught you that this is anabolism. We are going to see this pathway properly. Okay, we're going to see this pathway properly. This is overview. Overview means you are talking about everything else. And I've chosen to put this pathway, okay, because I like it so much. Okay, so the assessment of this course, I'm repeating myself. The continuous assessment is 40 marks. The exam is 60 marks. So you have 3 tests here. A test for structures and enzymes and bioenergetics. You also have a test on now this one, the component we have started, metabolism. Another test for medical genetics. Okay, so this one can actually have 2 tests in it because it's quite bulky. The exam will comprise of everything. Okay, we already talked about this when you're just starting. So the continuous assessment, you have got labs 5%, test 1 is 12.5%, test 2 is 10%, test 3 is carrying 12.5. Okay, so that's the distribution, but it changes on the mark allocation depending on the course coordinator. So this course is just to give students an insight into the biochemical basis of a wide range of clinical diseases and conditions on the world. So and it requires the application of molecular biology to medicine. So what's the aim of this course? It is going to introduce you guys to normal biochemical processes, and also it will make you understand how abnormal processes can result in diseases, and also how metabolism changes in pathological states when we are sick. Okay, when how does metabolism change when you are sick? And how do those abnormal processes result from normal biochemical processes? You have seen, I've given you an example of lead poisoning. Lead poisoning blocks this enzyme, and if this enzyme is blocked, you have increased ALA in the blood, which is going to accumulate, goes to the brain, and causes neural problems. And then the other thing is the hematology. Here you can see that the heme is not produced, or it is it's insufficiently produced. Right. So the books that we are using, there's a blue book that we are using. These books are just for supplementation, supplementary. So the books that if you want supplementary books, it's learning guys, okay? But there's a blue book which us we are using, the AC Academy students are using. So we have already talked about the biomolecules. We have carbohydrates. You can see the structures. We don't care about the structures now, but you must have the knowledge of structures, because you can't understand how metabolism is happening if you don't understand the structures. Okay, we have proteins. You can see the amino acids. All these things we have already done. Lipids, you can see we have already done lipids. Okay, so this is a person. This is bread and butter. I've already said that metabolism is the sum total of the anabolic reactions and catabolic reactions. So the sum of many interconnected reaction sequences that interconnect cellular metabolites is what metabolism is. Okay. So each sequence is regulated so as to provide what the cell needs at a particular time, and to expend energy only when it is necessary. The body doesn't just waste energy. No, waste things, waste products. No, it doesn't do that. It makes sure that it does what it's supposed to do at a particular time. So what is the importance of metabolism in medicine? We know that metabolism is required for the generation of energy, and the muscles are not going to contract if you don't have energy, because you need ATP, okay, to supply the phosphoryl group to the enzyme calcium ATPase. Okay, so you need metabolism for the generation of energy. You need metabolism for the synthesis of biological molecules, which is anabolism. We have so many biological molecules, and heme is one of them. Okay, hemoglobin. You need to understand that we have both catabolism and anabolism that have got unique features, and we are going to see the differences in a few minutes. Here you can see the compartmentalization of metabolic reactions. It means that we have reactions happening in the cytosol. We have reactions happening in the mitochondria. Okay, we have reactions happening in the smooth endoplasmic reticulum. We have reactions happening in the nucleus. We have reactions happening in the ribosomes. Okay, reactions everywhere. We have reactions. Okay, and those are appearing as pathways, either catabolic or anabolic. So this is just anatomy and PGY, which we already know. We're not going to dwell much on that. So we have already talked about anabolism and catabolism. Now we can go into the details of catabolism. So here we are breaking down a molecule, and mostly catabolism is associated with oxidative degradation. For example, the breakdown of glucose to produce pyruvate in a 10 series of reactions is catabolic in nature, is oxidative in nature. That is involving oxidative degradation of complex nutrients. For example, carbohydrates, lipids, proteins, and nucleic acids. Okay, to simple molecules, for example, lactic acid. Lactic acid can come from glucose, okay, via anaerobic respiration. We also have ethanol, carbon dioxide. These are molecules that are produced from complex breakdown of these molecules. Ammonia is one of them. You saw in you saw in the heme biosynthetic pathway, you saw that ammonia was living here. You see that ammonia is one of them. Okay, even water is one of them. Carbon dioxide. Okay, so it was a good thing to include this slide in the notes. Okay, and also it involves the release of what? Of energy. Right. So these reactions are hexagonic in nature and they release energy which will be captured as ATP or it will be conserved as energy rich electrons in the coenzyme molecules we call NAD plus and NADP plus. So these molecules they can get that particular energy and become NADH, and this one can become NAD pH. Okay, so which means they are carrying those energy, they are carrying the energy, they become high energy compounds, and then they go to the electron transport chain to go and release that energy in form of ATP. So we do have catabolic pathways that converge to a few end products. For example, water, carbon dioxide, and ammonia. We do have such catabolic pathways. Okay, so this one is something you need to know. It's a group of pathways here. You can see we have carbohydrates, we have proteins, and we have fats. So this is showing you the stages through which ATP is formed. How is ATP formed? For example, from glucose, from amino acids, and from fatty acids. So from glucose, you are going to pyruvate, but don't think this is just one step. There are so many things happening here, and this is called the glycolytic pathway, which we are doing next week. So glucose will be converted to pyruvate. Other things that can be converted to pyruvate are amino acids via transamination reactions. Okay, you can also convert amino acids to acetyl coenzyme A, and acetyl coenzyme A can also be formed from pyruvate. You can see that once we do deamination, we can form deamination of amino acids can give us acetyl coenzyme A. Even fatty acids can give us acetyl coenzyme A. Therefore, this molecule here, acetyl coenzyme A, is an amphibolic molecule. What does that mean? It means that it can go through a catabolic pathway or an anabolic pathway because it's the main thing. Okay, so glucose converted to pyruvate, pyruvate converted to acetyl coenzyme A, and acetyl coenzyme A can enter the tricarboxylic acid cycle, or better known as the citric acid cycle. So the tricarboxylic acid cycle is going now through a series of reactions. There are a lot, at least 8 reactions or 10. They are going to generate FADH2, which is required to enter the electron transport chain, and NADH, which is also required to enter the electron transport chain. What you are seeing down here is the electron transport chain. So this is step 1, where you are converting glucose to pyruvate. Then step 2 is converting pyruvate to acetyl coenzyme A. Step 3 is to convert acetyl coenzyme A so that it becomes NADH and FADH2, nicotine adenine dinucleotide in the reduced state. This is the ender another pathway called the electron transport chain, which is ETC. The electron transport chain now is going to generate ATP from ADP. You can see via the enzyme ATP synthase. Okay, and that is the final stage. So these are stages. Okay, and in this case, oxygen is reduced to
Sorry, sir.
water. You're going to see all these are topics. Yes, please.
I can't see your screen. I don't know if it's just me or even others.
No, it's just
I
you.
can see.
Okay, so these are topics. Don't think it's just one thing here. These are topics. There are a lot of topics in carbohydrate metabolism. There are a lot of topics under amino acid metabolism. There are a lot of topics under fatty acid metabolism. Okay, all of these they can converge and go to what? To the tricarboxylic acid cycle. This is an amphibolic pathway because it can it is a route where you can enter from anabolism, from catabolism, you go to anabolism. You see, you're coming from catabolism, but then you're getting anabolism down here. This is an amphibolic pathway. Okay. So anabolism, so in anabolism we have complex biomolecules that will be synthesized from simple precursors. Okay, for example, you saw that there was tryptophan can be converted to niacin, which is vitamin B3, a complex compound. So it is an energy requiring process. You need energy to form things, okay? Okay, you need energy to form things, which involves the formation of covalent bonds. And what is the source of this energy? The source of this energy is ATP or NADPH. Hey, you remember I've talked about amphibolic pathways. So what is an amphibolic pathway? So amphibolic intermediates will play a dual role, meaning between catabolism and anabolism, they are going to play both. So remember that certain pathways in the human body, for example, the citric acid cycle and also metabolites of other pathways, they serve both in catabolism and anabolism. Therefore, the citric acid cycle is an amphibolic pathway. So you have to remember that. So this is the summary. You can see catabolism this side and anabolism this side. We can make acetyl coenzyme A from this amino acid isoleucine, from leucine, from pyruvate. So pyruvate can be made from serine, which is another amino acid, or from alanine, or from glucose. Glucose can be made from sucrose. Okay. Triglycerides can be broken down to fatty acids or phospholipids, which can make acetyl coenzyme A or an amino acid called phenylalanine. Okay. In anabolism, you can see here we can make things like the biosource or the steroid hormones or cholesterol from acetyl coenzyme A. You see, acetyl coenzyme A is going to become acetyl CoA, which is going to become mevalonate, which is going to become isopentyl pyrophosphate, which is going to be cholesterol. Then cholesterol can become biosalts and steroid hormones, the steroids. Okay. You can also have fatty soluble vitamins from this compound. You can also have carotenoids, okay, like vitamin A. Okay. Acetyl coenzyme A can also form the fatty acids. Okay, that's the reason why when you're eating too much of glucose or carbohydrates, you become fat because of this. Glucose is converted to pyruvate to acetyl coenzyme A to acetyl coenzyme A to fatty acids to triglycerides. Because the differences between catabolism and anabolism. So catabolism mostly it involves the decomposition of large complex molecules into smaller ones. Okay, but anabolism you are breaking down the large complex organic molecules. Sorry, you are making the large complex organic molecules from the small molecules. You should also remember that catabolism is degradative, meaning it's breaking down, whereas anabolism is biosynthetic, meaning students should now start using technical language, not say anabolism or you're making. No, it's biosynthetic and degradative. Okay, so it's either a pathway is biosynthetic or degradative. Please keep that in mind. Catabolism is oxidative, whereas anabolism is reductive. You know what that means when you're transferring the hydrogen. So meaning you are removing hydrogen from things in oxidative, in catabolism. Catabolism is going to give you energy, but anabolism requires you to put in energy. And the catabolism is converging, whereas anabolism is diverging, meaning catabolism is going to reach to one thing, whereas anabolism is going to reach to so many things. Okay, and this marks the end of today's class. Any questions? Any questions? Hello? Have I lost anyone? Nivea, you don't have questions for me?
For now, no, sir.
Are you happy with this class? Who is happy with this class? Raise your hand if you are happy with this class. Because it's kind of like the starting point of everything. Okay? Okay. So people are happy. If you have any questions, please ask. The test will be on Monday. So test is on Monday. For this course, the test is on Monday. So we are supposed to do a series of revisions. More I'm not teaching. So we can do a revision at 14 hours. Even on Thursday I'm not teaching. Even on Saturday I'm not teaching. Even on Sunday I'm not teaching. Right. So the test will be 14 hours because we don't want to interrupt the learning process. We want to continue with the learning while people are writing assessments. Okay, so if you don't have questions, I'll stop my recording because I don't want to continue recording. The class has ended. We don't go beyond this in this class. This is an overview. Okay, the group is open for questions. You can